Yeah, so my name is Philip Kiser. I'm an associate professor in the Department of Physiology and Biophysics. I have joint appointments in the departments of ophthalmology and clinical pharmacy practice. And my laboratory studies the roles of retinoids and carotenoids in vision. So we have several projects ongoing right now focused on understanding the biochemistry and the structural biology of the visual cycle pathway, and I can just quickly describe that. The visual cycle is a metabolic pathway that regenerates something called a visual chromophore. It's a vitamin A derivative called 11-cis-retinal for the light sensors in our eyes. They're known as opsins or visual pigments, rods and cone visual pigments. And so we spend a lot of time studying this pathway because it's of very high importance for human health. If the enzymes or other proteins in this pathway are non-functional, they can result in retinal diseases. And on the other side of the coin, in certain retinal diseases, overactivity of this pathway can drive the disease pathogenesis. And we've done studies looking at both, trying to understand how mutations lead to protein dysfunction, as well as ways that we can modulate that pathway to protect the retina in certain conditions like Stargardt disease and age-related macular degeneration. So one project that is ongoing in the lab right now that kind of is a very collaborative project involving basically everybody in the lab is a study where we're looking at the role of a specific protein-protein interaction in the function of the visual cycle. And this protein-protein interaction involves a retinoid binding protein called cellular retinaldehyde binding protein. It's the last protein of the visual cycle that accepts visual chromophore and shuttles it through the RPE so it can be delivered to the photoreceptors. And there was a while ago a protein interaction that had been identified that no one really followed up on. And we've been able to generate a new mouse line where we're able to directly test the importance of that. And this kind of gets at a bigger picture question about the roles of protein complexes in visual cycle function. So in terms of breakthroughs in the field, I think that there's a few different points I could make on this. I'm on the sort of basic research side of things, and then some others on the therapeutic side. On the basic research side of things, I think we for the most part know the individual protein components of the visual cycle, but really understanding how they work together to ensure that the visual cycle functions with efficient kinetics is sort of the next frontier. And I think that there's kind of new technologies that have been developed over the last few years that make this time very ripe to go back and start looking into those. So that's one of the directions that we want to go. On the therapeutic side, the next, I think, frontier in terms of the visual cycle modulation concept that I discussed earlier would be to really come up with compounds that can have fewer side effects. And it's really been the side effects of these medications that have prevented their advancement into becoming FDA approved. And then finally, something that we don't directly do in the lab, and I think it's a very important next step in this area, is the development of gene editing, base editor technology to correct mutations in visual cycle proteins to basically become sort of a general way to fix some of these genetic problems that lead to blindness in children, for example.